talk to you, I want to see what happened. If you didn't get a score that you liked, I want to work with you individually to see if we can figure out why. And then we'll make a plan for something. And for some of you, it may involve like sitting down, it may happen at the same time that we talk about what's going on. We may talk about some content stuff, and if, for example, it becomes clear to me in our conversation that you know this stuff and you know it really well, but you just couldn't quite get it on paper, then we might be able to just chat and like I can adjust your score through what is officially known as a verbal exam. Like we can do that. Others of you, I may say, okay, it seemed like you understood this and this and this and this. This piece you're a little weak on, go do something and then give it to me later. And that will be something where you learn and then show me what you've learned. So that might be, you know, like if you struggled with the lactase question, the FRQ question, it may have you do some more detailed research and kind of report back so that, because what we really want is for you to have a really solid understanding of what lactose is, what lactase is, how enzymes work, how denaturing can happen, and how we would measure it. Like all of that stuff is what we're really looking for. And for some of you, it may be that you, like, I've had a couple conversations with people already where it's pretty clear that, and this was a good conversation, don't get me wrong, but it was pretty obvious that what happened was they just hadn't really prepared. And so for them, I said, go prepare, and then you just take it again. Take the same test again. You max out at 80%, which is not as good as I think what most people would like, but it's better than what they got to begin with. And if the take-home message is next time, make sure you're really prepared, then great. So what I want is for you to connect with me as soon as possible if you're wanting to do a retake. We'll start with a conversation, and what happens next depends on what we discover during that conversation. Okay, any questions about that? Okay, so here's where you get to see my artwork. Are you excited? The next activity that we're going to work on, we'll do part of it today and part of it on Monday, is an activity where we're thinking about cell size and shape. And actually we're not going to be working with actual cells because they're too small to see. So what we're going to do is we're going to use potatoes as a model for cells, and we're going to vary the size and shape of the pieces of potato that we use. And what we want to investigate is how does the size and shape of potatoes, like chunks of potato, affect the rate at which diffusion occurs on the outside. And we haven't talked about diffusion yet. We will next week. But most of you have a pretty good working idea of what diffusion is from previous biology classes. It's just the movement, random, spontaneous movement of things from one place to another. That's it. So what we're going to do is we're going to take our chunks of potato and we're going to put them into an iodine solution. And the iodine will diffuse into the potatoes. Iodine will react with a complex carbohydrate in these potatoes called starch to turn the potatoes purple. Wherever the iodine meets starch, it'll turn purple. So what we're going to be able to do is see how quickly the iodine is moving in by seeing how much of the inside part of these little chunks of potato turn purple after like an hour or so. So here's the setup, and here's my artwork. I can tell it. This is one of the things I love about this class is the sense of eager anticipation when students know that I'm about to do artwork. Because if you haven't heard it, I'm a fantastic artist. This is all going to be photorealistic. So here's what you're going to do. You're going to start with a potato, and potatoes are plants. So this green blob represents a potato, which you already knew because it's just amazing work. So here's our potato, and you're going to cut. I have several potatoes, so you're going to have to work in probably groups of 3 or 4 to do this. But what you're going to do is you're going to cut it up into several different chunks. Okay, you're gonna cut it up into 4 cubes, and those 4 cubes should range in size from about 1 centimeter on a side, isn't that an amazing cube, to about 2 centimeters on a side. That's better. Okay, and these will all be the same shape, obviously, but they'll have different volumes. And it doesn't matter exactly how big they are as long as they're different. So it's somewhere between 1 and 2 centimeters on a side, but they're all cubes. The other thing that you're gonna do is you're gonna cut the potato into 4 shapes that are roughly the same volume, about 6 cubic centimeters. So you might have to do just the tiniest little bit of math here, but 4 different shapes. So for that, you're gonna make 4 shapes that are all about 6 centimeters cubed. That's the same volume as this shape. No, it's not. This is 2, 4, 8. Let's do, well, 6 is fine. So roughly 6 centimeters cubed, so somewhere in between these 2 shapes in volume. And one of them will be a cube. One of them will be a rectangular solid, and you can make the proportions of this whatever you want as long as the area matches. One is going to be a sphere. You ever seen a better sphere? And one of them will be a cylinder. And again, the relative proportions of the cylinder can vary. The cube and the sphere should be like as close as you can get to a perfect sphere and a perfect cube, which isn't going to be very close, but it'll be good enough. Okay. So once you've cut these shapes, you're going to make. So as you cut them, you'll kind of have an idea of how big you're trying to make them. But once you've cut them, you want to measure whatever you need to measure to calculate surface area and volume. So let me be clear, I just wrote measure surface area and volume, but you're not measuring surface area and volume. You're measuring lengths. Measure the length of the sides, measure the radius, measure the height, so that later on, it'll be easy for you to calculate the surface area and volume of all these shapes. Yeah?
One thing I'd like to get straight between the shapes and stuff. Are
Yeah.
we supposed to make 4 cubes different sizes?
Correct. 4 cubes of different sizes ranging from 1 centimeter on a side to 2 centimeters on a side. So they'll all have different volumes and surface areas, but they'll all have the same shape.
And then all of the weird shapes are supposed to be 6 centimeters.
Roughly, yeah. And what you'll find is it's not gonna be perfect, but
Okay.
it'll be close. And we've got the formulas, so you'll be able to kind of plug some numbers in to figure it out. Yeah.
What are we using to cut them?
You are using Maliahi brand knives. These are not Maliahi brand knives, but these are from her house. And basically what's gonna, you have 1 Dan Briegar brand knife too. No, 2 Dan Briegar brand knives too. This is my knife. And what's gonna happen is that you'll make your initial like rough shapes with those knives, and then you're gonna carve them to size with these official piece of junk miscellaneous butter knives that we found somewhere. And I know that sounds crazy, but actually it'll work, because if you take a piece of potato, you won't be able to get good like straight cuts with this, but once you cut it, you can kind of shave it pretty easily to turn a cube into a sphere, or a rectangle into a cylinder, or a cube. Yeah, so you'll be able to make edits with these butter knives. This is not ideal, but this is what we have. All right, so then this stuff is all going on, and in addition to that, you're gonna prepare a beaker, maybe 2, but I think you can probably do this with just 1. So you're gonna take a beaker. Here again is an absolutely fabulous drawing of a beaker. They are 500 milliliter beakers, and what I think is going to work best, although we may find out as the period goes on, we may find out that we need to make some adjustments to this. But what I think is going to work best is if you put 50 milliliters of iodine solution in there. That's from here. 50 milliliters is gonna be to the first tick mark right off the bottom. It's not much, and that's what we want. We don't want much. And then you'll add 450 milliliters of water to bring it up to a total of 500 milliliters of liquid. This is gonna go on one of our hot plates, but we don't actually wanna get it hot. We just wanna kind of warm it up to about 30 to 35 degrees. It'll diffuse a little bit more quickly. So we have a bunch of thermometers, and you won't leave the thermometer in your container the whole time, but occasionally, as you're doing other things, you'll check the temperature. And you wanna try to keep it between 30 and 35 degrees Celsius. That, I think, will give us a reasonable rate of diffusion. If it gets too hot, you can either take your beaker off temporarily or turn the hot pad off depending on how hot it's getting. And if it's not warming up enough, you can turn it up. It doesn't have to run at it doesn't have to be, we're not comparing results from different groups. So if your potatoes are sitting at 35 degrees and somebody else's end up at 41 degrees, it's not gonna be a big deal. You're looking at your data and that's it, okay? This is the setup. This is the part that needs to run for about an hour. And then after that hour is done, then we're gonna get the potatoes out, slice them in half, and make some measurements. And so what I need from you is to just kind of like use your time in between having this all set up and soaking and an hour from then to do two things. The first is those interviews for the cell structure activity that we started on Wednesday. So ideally, you were able to get your research done on Wednesday. So you and your partner had a structure, and you should be pretty well versed in where it's found. And when it says what type of cells, that could just be, for some of these, it might just be eukaryotic, prokaryotic, or all cells. For some of them, though, it might be more specific than that. Like if you had chloroplasts, those are only found in photosynthetic cells. And so those aren't going to be in animal cells or fungal cells. And then the function of the structure, what process the structure is involved in, and how it interacts with other structures. So you should have this information. And then while your potatoes are soaking, I want you and your partner to go find other people and their partner and talk to them about what they discovered for their structure. And it shouldn't take more than maybe a half an hour, 45 minutes for you to talk to all the other groups and get to the point where you have information written down for all of the cell structures that people were studying. This you want to take a picture of and turn in on Canvas when you're done. This is due today. And then after you've got that done, if you have time, which I think you will, but I'm not 100% sure, you'll still be waiting for your potatoes. They'll still be heating up. So the next thing you can do is you can do some of the calculations that are part of the assignment. So this is posted to Canvas, and if you click on the assignment, it says what was the total volume of your pieces of potato, the total surface area, and then the surface area to volume ratio. So you have a bunch of calculations to do, none of which is particularly complex. All the relevant formulas are on here. So you've got volume of all four of the shapes that you'll be using, surface area of all four of the shapes you're using. Round to your heart's content. Pi, by the way, is 3.14159 something. Just use 3. Like these are tiny little pieces of potato. Pi is 3 for this experiment. Make your lives easy. And round your measurements too. Like if you measure something and it's 11.71 millimeters, that's 1 centimeter, right? Like just keep it simple for yourself. Okay, that, my fine feathered friends, I think is enough to get you started. Francis?
Let me get this straight. We're supposed to be doing potatoes and then interviews and then back to potatoes.
Yeah, so potatoes, interviews, calculations, and then there's going to be some measurements with the potatoes, but I'll tell you how to do that in about an hour. We'll talk, yeah. Yeah.
How did you just do the calculations if you haven't done the measurements?
You have. Sorry, let me be more specific here. When you cut your potatoes, make the measurements that you'll need to make. Hey, everybody, you should listen, just to make sure you've got this, okay? You should make any measurements that you need to make to calculate the surface area and volume of the pieces of potatoes. Those measurements come first. When you do these calculations, what it says in the module is make the first 3 sets of calculations. So that is the total volume of the potatoes, the total surface area of each piece of potatoes, and the surface area to volume ratio. The next calculations you're going to do require you to measure how far the iodine diffused into the potato, so you can't do those yet. Does that make more sense? Okay. Anybody else have any other questions? Okay, you can cut right on the lab benches. Just be gentle. Like you can just use a knife on them. I have 1 5-pound bag of potatoes, both for you and for the other class. So like I said, you're going to need to work in groups of 3 or 4, and you probably are going to have like a half potato each. I think is like that'll be a realistic amount of potato to use. So if you need to modify the sizes, like you can make things smaller or bigger. You're only comparing your data to itself. I don't think I need to tell you to be careful with the knives. I've got beakers, thermometers, and hot plates there. I think that might be it. Does anybody have any questions? Okay, let's play with potatoes and see what we get.